Hello, I'm Lisa Pointner from the Department of Medical Biology and Biosciences of the University of Salzburg. I'm pleased to give you an insight into our study in which we investigated whether birch pollen can directly influence the IL-4 receptor interaction. Birch pollen is a clinically relevant allergenic source in Europe. Allergic sensitization is orchestrated by an aberrant Th2 immune response. The key cytokine inducing this process is IL-4, acting via the type 1 IL-4 receptor composed of the IL-4 receptor alpha chain. The initial signals inducing Th2 differentiation by birch pollen are still unclear. However, as the pollen context has adjuvant properties, could the initial IL-4 signal originate from the birch pollen source itself? To find clues to this question, we investigated the interaction between compounds of a birch pollen extract with the IL-4 receptor alpha chain. We found that birch pollen can bind to IL-4 receptor alpha in a dose-dependent manner, comparable to the natural IL-4 ligand. In contrast, neither ragweed nor grass pollen extract displayed this binding activity, indicating a birch pollen-specific effect. Protein degradation reduced the binding activity, suggesting a protein ligand. To further characterize this ligand, we generated fractions of different sizes derived from the birch pollen extract. Only the high molecular weight fraction retained the IL-4 receptor alpha binding activity. As the birch pollen extract is able to induce a Th2 response in an IL-4 reporter mouse model, we tested if the high molecular weight fraction also holds this feature. In fact, the in vivo Th2 inducing capacity of the birch pollen extract remained in the high molecular weight fraction. Our results present the existence of a birch pollen derived compound that can interact with the IL-4 receptor alpha chain. This property seems to be birch pollen specific, affected by protein degradation, contained in the high molecular weight fraction, and is associated with a Th2 induction. Whether we are dealing with a true IL-4 mimic remains to be further explored by investigating the downstream signaling mechanisms and the involved cell types in vivo. Our research is important to better understand the initiating mechanisms of birch pollen allergy and will provide novel therapeutic targets. Thank you for your attention.